much, Munir. So it sounds a bit ridiculous to have a diabetologist here talking about stratified medicine or precision medicine. We're not really known for that. We tend to spend all our time treating people that are pretty unwell and then dealing with their pretty horrific complications. Diagnosis, technology are not words that you associate with a diabetologist. And so really what I want to talk about is some specific areas. And I think really what we have come to deal with is the idea that stratified medicine is about omics, it's about technology, it's about defining its deep phenotyping. And in some ways there are areas of diabetes where that's true. This is Lily. She was diagnosed at the age of 3 months, desperately ill in ketoacidosis, and was diagnosed as having type 1 diabetes. But those of you will realise, as immunologists here will say, well, you can't have an autoimmune disease in the first 6 months of life. And indeed, you cannot have type 1 diabetes in the first 6 months of life. And if anyone wants to prove me wrong, I'll pay them £100 if they find a case. So we had a very nice clinical criteria which said this wasn't type 1, so it was going to be genetic. It was going to be a single gene disorder. And this was a case where we found the gene, which was a gene in the potassium channel, but the beauty was that there was 20 years of science which preceded this, which said that this channel didn't only close to ATP where the mutations were, but it also closed with sulfonylureas. And so that gave a possibility of a precision therapy for these insulin-dependent patients. It sounded ridiculous to give a sulfonylurea to a person who had no C-peptide, but foolishly we did it, and even more remarkably, it worked. And it didn't just get them off insulin in a kind of, they're doing okay, they did brilliantly. And actually, if you look at this graph here, you can see the HbA1cs went from the typical level of an insulin-dependent to close to the normal range without hypoglycemia. And that was because they now had this food, insulin secretion, that was directly related to meals. So this is a clear example. So if you want to do any trials on this, you've got a clinical group, subgroup that you take under 6 months. You then have a specific, you do genetic testing, you define a subgroup. There are ones where the biology says clearly they should have this therapy, and it works. Now, that's pretty easy, and that's what we're hoping to do. And really the early wins have come from this kind of genetic model. It's really cancer and monogenic conditions where the heterogeneity is easily defined. Now, in neonatal diabetes, there are 25 different causes, but 2 of them respond to sulfonylureas, and they're about half the cases. And the biology defined can help define the treatment. And the actual individual response, if it's big enough, that's going to be easily assessed. And so we've moved to this idea that this is how stratified medicine is. And I think then you start saying, well, hang on, Andrew, how many neonatal diabetes? And you say, well, it's 1 in 100,000 births. And I say, well, that's not there's not many of them. You do a lot for them, but there's not a lot of them. What about type 2 diabetes? 4 million people in the UK. How about sorting them out? Well, this is 5 years training for a diabetologist. You can do it a bit quicker than that. Basically, the higher the blood glucose, you either give them tablets, then you add another tablet, and then finally you give them insulin. It takes us 5 years to learn that. We're not the smartest doctors. But what that means is our treatment depends on the severity of the glucose. It doesn't depend on the etiology. And really, that's a real question. And obviously, any approach of precision diabetes, you would tailor it to the cause. And on the face of it, this looks a pretty good treatment to do. You know, if you're going to do stratified type 2 diabetes, phenomenal variation in response. Now be careful, if you've read the things that Stephen Sen wrote, you'll know to be careful to say that means you can stratify. But there is variation there. You've got a great biomarker, an HbA1c, looking at glucose control. You've got variation in physiology. We know some of our patients have a beta cell defect, others are much more insulin resistant. And then we've got 7 different types of drug that work in different ways. And there have been some initiatives. So it really looks like this is a good place to start. Until you start thinking about it, and you start to realize that these said diabetologists don't even know which treatment does better in obese against non-obese patients. 50% of our patients are not obese. We do not have any difference in guidance for them, although their pathophysiology is clearly different. And So you're starting to think, and so this is the definition we're using for this one, that it's a smarter model of medicine in which tools are used to stratify cohorts of patients by subclasses of disease or likelihood to response. So which tools has almost inevitably meant omics. And when you stop to think about it, which GP is going to send something off for DNA sequencing? And the other problem is, what tissue are you going to send? The problem is the pancreas. Very few patients are prepared to give up their pancreases to know what treatment they should go on for their type 2 diabetes. So, and then you've got this thing about measuring response. Now you say, well, we want you to stop people dying. If you've got cancer, it's pretty easy to stop people dying. If you've got type 2 diabetes, they take quite a long time to die. So you have to do massive studies. And for all these years, we've only finally got 2 studies that have shown an improvement in mortality. What chance of showing that in stratification when there will be smaller numbers? So what we did was we decided that really we couldn't do that. So what we would do is we were going to see is could stratification occurred by clinical criteria? I.e. start with the solution. The solution had to be a GP did it. We had to do information that the GP collected. And then we would decide that we'd do this initial work on hemoglobin 4. And this is the basic idea. You've got Anthony and Beryl, and according to NICE, they're both the same because they're both on metformin and they've got an HbA1c of 8.5% and they need a second therapy. But they're clearly not the same. They're different ages, they're different shapes, they will have different pathophysiology. And so it does make sense that clinical criteria could pick out different people. And really, as I say, our guidelines at the moment would just say, and not only do they say that they should be treated similarly, but they also give you absolutely no clue which one you should do. After metformin, there are 4 therapies available. You can use them all. That summarises what NICE decided. They did initially say which ones had the best evidence, which were cost effective, but the pharmaceutical industry advised them not to say that. So that's where we are. And so you've got a complete blank sheet. And we were extremely lucky because we were given money by the MRC to allow us to try and develop something. We had to develop something new and something different. And what we did was get a bunch of people that thought differently, really, because there wasn't a department of stratified medicine in type 2 diabetes. We also were extremely helpful to have industrial colleagues who came along and actually gave the crucial bits of insight. So this was our approach. Our approach was that we would develop a model using routine data, that we would say, let's see if these clinical factors actually do change who responds and who doesn't. Now, obviously, there's all sorts of problem with routine data. What you want people is to be randomly allocated to drugs. So therefore, we wanted to take the model we developed in the routine data and test that within trial data. Now, that was a real problem when we started because we needed individual level data. And whoever we spoke to in the drug company, there was always somebody that said no. But all of that has changed, and now with the availability of trial data, that made this a real stage. And then we've got a final stage that we won't talk about, which is actually using crossover studies, which you can use in chronic conditions. So this was our idea. So how, if we're using routine data, how are we going to dope with the fact some of the manufacturers said, well, our drug works in everybody except for those who don't take it. And how are you going to stop it just being a concordance thing that you're studying? How are you going to measure the response? NICE has 2 ways of saying response. One, it says how much does it fall, and some drugs it must fall 1% before you actually allowed to carry on using them. But sometimes they say you must reach a target. Which of those responses were we going to use? And we'd get very different people. And really, were the clinical subgroups going to be robust? Was every GP going to put people into the same subgroup? Because if they didn't, it was pretty useless. And there was this big thing originally with the availability of the data. After that, the issue came more that drug companies would tend to use the worst drug to compare their drug against when they did the comparative trials. So that's a bit of an issue. And finally, the design, I think, and there are other questions there, but I'll leave that. So the first thing that Andrew Farmer did from Oxford was to say, well, can we actually get rid of the people who don't take their drugs? And the way we decided is, well, let's look at response. And what we're able to show was that if your prescription coverage was below 80%, you didn't get the response that was seen in people that had prescription coverage above that. So at a practical level, we're going to say, we're going to discard that data and we could take prescription coverage from routine data. So we got rid of those. The next thing was response, and it's not the brain of Britain kind of stuff to work out that the higher HbA1c, the more it's going to fall. And the biggest predictor of, actually with all conditions, in all these stratified things, of who does best, if you're looking at a size of fall, it's the people who start highest. And that happens in every single condition. And what that means is that you can't just, and because your data is no longer randomized between two groups, you've got differences in starting HbA1c, which might be systematic. The obese might start off systematically with a higher HbA1c. And actually, after a lot of discussion, the best thing we did was to look at the initial HbA1c fall and correct for the initial HbA1c. And so those were our approaches. And so the real question was, does it work? And so CPRD is the most fantastic NIHR resource. And this was data, we were now looking at 250,000 people with type 2 diabetes at the cost of CPRD gold. I mean, that is fantastic. And what we were able to do was look at some things like, well, does the age of diagnosis make any difference to response? And it does make a difference. You do far worse if you're diagnosed younger, but that's with all therapies. So if you're trying to do stratification, that's not going to work. That's not going to help you choose which one. It just means if you've got a young type 2, they're going to be very hard to treat. But some show differences, and this was the question of obese versus non-obese. And all therapies did worse in obese patients, including metformin, that we traditionally think is good in obese patients, except for the thiazolidine diets. But the other interesting data was then you could look at things like gender, something that most GPs could accurately subclassify patients into. But there was a real difference. Sulfonylurea, and we didn't know this, sulfonylureas did much better in men, and thiazolidine diones and metformin do better in women. And that meant we could start to stratify the population into 4 groups based on obesity, which roughly divided by half, the present, and male and female. And what you can see there in the routine data, this showed clear differences in HbA1c response, and the difference between the 2 drugs in these 2 groups was about the same as another drug, which is pretty clinically significant. So then obviously you have to worry, this is routine data, what about when people are randomly assigned? And so we're able to go back to the drug trials and redraw them using these subgroups. And this is the ADOPT trial, and there basically are 2 versions of the ADOPT trial. The ADOPT trial if you're an obese female, and the ADOPT version that when you're a non-obese male, look completely different. And so you can see in the obese females, we have the thiazolidine diones doing phenomenally well. They keep their HbA1c down for the whole 5 years, whereas the sulfonylureas rapidly deteriorate. So clearly, in terms of glycemic response, and in contrast, in the non-obese males, the sulfonylureas do better initially and remain better until 4 years. So what sense is it to show people the group data? Now again, that's only 1 trial, so we then did it in another trial, which is the record trial, and again we saw the same subgroups. So we're able to see again this response to non-obese males, which is now up there. You see the sulfonylureas doing better. The obese females down here, you see the thiazolidine diones doing better. But also we could then divide the subgroups, the side effects, which is the other key thing. And actually we saw clear differences here in things like fractures, which really were not found in the males, and edema was hypoglycemia occurred in all the groups equally. It wasn't altered by the response. And so what that means is you can start to stratify at a very simple level. So really, in conclusion, that different diseases need different approaches. There is not a stratified medicine approach. And the most crucial thing is to define it about where you're going to go ultimately. If it's a GP disease, it's got to be defined in general practice and doable there. And not all stratification needs to involve omics. Now, we could have been up here talking about neonatal diabetes, fascinating omics, but it's not dealing with a major health problem. I think a lot of these big things, we're going to have to use a lot lower tech approach to that. And clearly defined subgroups within diabetes can get differential responses with different therapies, even if they're just whether they're male or female and obese or non-obese. And really it's been the massive data sets and the availability of trial data that really are crucial new tools for defining stratification. Thank you.
Thank you very much, Andrew. That was fascinating in terms of using clinical stratification. Any questions for Andrew? Go ahead, Eddie.
I noticed you had Andrew Morris on your group of collaborators. Andrew famously had a database of diabetics for about 10 years or more. Did that database, which was collective?
Can you press your button for the microphone?
I think it's on. Yeah. So the question was about Andrew Morris. He had a database of 10 years of, I guess, retrospective data. He's now using that data prospectively. Have you been able to use that in your studies as well to gain info? I guess my question is, does that retrospective data add anything to what you want to do prospectively?
So, I mean, the retrospective data is great because if you want to look at the 5-year response, otherwise you have to wait 5 years for it. I think the advantage of when you can link it to the patient, or you can do it prospectively, you can then actually design trials going forward. And I think that's the next thing to say. We've looked at that in this situation. I think one of the things is it doesn't have to be so complicated. I mean, really, the difficulty for Menier is because he needed a sample from the patient. And if you just think, is there things we could do without needing a sample? Because all of those difficulties came because he needed DNA. I mean, there are advantages in Scotland and in Tayside, and Ewan Pearson doing great work there. And he's got the advantage that he's got DNA on all the patients already. And so that is, and Biobank is the same, and there are phenomenal advantages that come from that. But I still think that's not actually going to come into the general practice clinic. So I think it's important. I think prospective, I think we need to go on looking at it. I think this is a very real example of how that data and the trial data can be used without it being really that difficult or complicated.
So Andrew, if you want to close the loop in terms of a clinical stratifiers, I guess the next stage for you is to get NICE to approve it, and then for GPs, etc., to use it and to see whether it truly makes a difference in clinical outcomes. So how far are you along down that road?
Yeah, and that's difficult. Obviously, the first step is you have to persuade authors editors that they wish to publish it. And I think one of the things is if you're doing something that is iconoclastic, that is questioning previous things, then it is quite hard to get that out there. So in terms of NICE, I think the regulators are a crucial part, and I think one of the things we're doing is working with the health technology and really asking what evidence would you want? It's always possible to say, well, you haven't got hard endpoint, but we've lived without that for the last 30 years, and to be honest, if they want hard endpoint, we're not going to get it. But most people are giving a glucose-lowering drug to lower glucose. That's their main role. So we hope that we'll be able to. We're in discussion now.
Okay. So I think that's another important point that perhaps we need to look at this afternoon as well is what kind of evidence do you need to take precision medicine into practice, as part of the overall pathway for research?
And I think one of the questions which none of the trials that I heard about were answering is it better than a non-stratified approach? Yeah. I.e. you give everybody the same drug, and those trials are going to be very hard to power appropriately.
Okay. Well, thank you very much, Andrew. That was great. Thank you.